Hi everyone, today is part 1 of part 2 of the microscopy series. I just thought that there's too much content to fit into one episode, so let's get started. There's most likely going to ask you to convert units, so you just have to know these really. There's only 3. So this stands for millimetres, you'll come across that from centimetres. This is micrometers. You might not recognise that symbol. And this is nanometers. Because micrometers are smaller than millimeters, we have to times by 1,000. And because to get from this stage to this stage, you times, the opposite of that is going to be divide. So if you memorise that getting from that to that is times, then you can just remember that the others divide because it's just the reverse action. Okay, and I think this is a neat little remembering method for the calculating magnification, and all you've got to remember is I am. So I stands for image size, the A is the actual size, so that's like the real size of the object. Image size is just the one that you can see from your microscope. Magnification is how much it is enlarged by. So if you wanted to calculate actual size, you'd cover up the A, then you see that I over M, so divide image size and magnification, and that works for all 3. Thing that's going to be above the stage here. So this big rectangle, it'll be see-through, but this is the slide. This is just a cover slip, and in the next lesson we will learn how to position it, because you have to position it at an angle in order for there to be no air bubbles in it, and it's important that we avoid the air bubbles so we can get a clearer picture. And here's a specimen, and we put a drop of water onto the cover slip and an iodine, and the iodine helps us to see the subcellular features of the specimen. And the person that discovered this method was Robert Hooke, and he discovered that staining things with iodine helped us to see them. Okay, so we're going to look at the differences between the light microscope, which is the one I showed you earlier, and the electron microscope. So firstly, as we saw before, it uses light to form an image, but the electron microscope uses electrons, which is indicated in the name. These electrons are bounced off against the specimen, and this results in high magnification and high resolution. Now let's look at the definitions. So magnification is a measure of the size of an image compared to the size of the object, so basically how much it's enlarged by. Resolution, the ability to distinguish 2 objects from each other, which is basically how detailed the image is. And light microscopes are a lot lower in magnification and a lot lower in resolution. So because of this, you can only see, you can see cells, but the larger subcellular features like the nucleus and the cell walls. Electron microscopes allow us to see a lot more smaller subcellular features, and even as small as ribosomes. And this has led to a lot of scientific advancements in learning the parts of the cell. Light microscopes are the ones I use in schools, but electron microscopes are a lot more expensive and larger than light microscopes. So we're going to look at some exam questions. We'll just look at the first one. Cells can be visualised with a light microscope. Figure 2 shows some smooth muscle cells from the wall of the small intestine. We looked at the beginning of this question in our last video, so go check that out if you haven't. But this is just a different question from the same one. Suggest why it is possible to visualise mitochondria using a light microscope, but not ribosomes. Okay, pause the video and jot down your answer. Okay, mark your answer. So we've just learned that light microscopes have nowhere near the amount of magnification or resolution than electrons, so they cannot see smaller subcellular features. Question 2. Figure 3 shows a bacterial cell and an animal cell. The cells in figure 3 are drawn to a length of 100 millimeters in the student's textbook. The actual length of the animal cell is 60 micrometers. Calculate the magnification of the animal cell to two significant figures. Show clearly how you work out your answer. So remember to put the calculation, to write that down for you to get all the marks. I think this would probably be more than 2 marks, to be fair, because you have to convert it to significant figures, and remember there's some conversion here, so pause the video. Okay, you wouldn't have got anywhere if you hadn't have converted this 60 micrometers into millimeters, and then they've plugged it into the equation, so they've written the equation here, now they've plugged it in, calculated it and converted to two significant figures, and it highlights here that you need to convert. Okay, last question, I promise. The nucleus of an animal cell has a diameter even, of about 6 micrometers. Suggest a reason as to why bacteria do not have a nucleus. Okay, so have a think about this and pause the video. Okay, mark your answer. Bacterial cell is too small. The nucleus is larger than a bacterial cell / no room. Okay. Thank you for watching. If you have any questions, I'll be happy to answer them. Take a break, and thanks for watching.